Yes.
Yes.
Okay. Last time we talked about the excretory system of human beings, and we broke it down into 3 broad categories. We started with the large excretory system, including all the things that are consisted in the excretory system, where we have 2 kidneys. We have the urethra, you have the ureter, you have the urinary bladder, you have the ureter, the kidney, the bladder, the kidneys, the aorta, and the vena cava, the renal arteries, all those parts. Then we say the essential organ in the excretory system, or the functional organ, is the kidney. The bladder is important, but the bladder is just a collection point. The functional organ is the kidney, where we have the kidney. That is the structure of the kidney. We have there the cortex. This is the cortex, and then this is, make this. The cortex is this one here. We have the cortex, the medulla. The medulla, those pyramid-shaped organs, pyramid-shaped parts. These are what we call the medulla. Oh, this is small. Just a moment. Yes. So this is the cortex. This part here I'm shading. I'm using the wrong color. This is the cortex where I'm shading black. And then this one here is the medulla. I'm going to shade it red. This is the medulla. So we have the cortex, the medulla, and then we have the pelvis. This is the pelvis. This part here is the pelvis. Then we have the renal artery and the renal vein. The renal vein carries blood away. The renal artery brings blood to the kidney. Then the ureter simply takes blood, so it takes away urine that has been filtered from the blood. We talked about that last time. I talked about the functions of the kidney to carry out excretion, osmoregulation, and endocrine glands which secrete hormones. Then we talk about the functional part of the kidney. Not every part of the kidney does filtration. The part of the kidney that does filtration is the nephron, and that is the functional unit of the kidney. The nephron is what carries out excretion in the kidney. So these are the parts of the nephron. I have a simplified one here. We have the glomerulus, the BOWMAN'S capsule. The BOWMAN'S capsule leads to the proximal convoluted tubule, and then we have the loop of Henle here that looks like that is U-shaped. That's the loop of Henle. Then we have the diastole convoluted tubule. I explained and said that the proximal convoluted tubule is all an English word. It's not a scientific word. The word proximal means nearest, convoluted means coiled. So we are talking about the coiled tube nearest to the BOWMAN'S capsule. That's the proximal convoluted tubule. And then the word diastole convoluted tubule is also entirely an English word to mean the coiled tube away. Diastole means away, convoluted again means coiled. So we have the coiled tube away from the loop, away from the BOWMAN'S capsule. Then we have here the collecting duct. These branches that you're seeing here indicate where other loop of Henle is joined, connect to this collecting duct. So that is the simplified diagram of the loop of Henle. And then we talked about the parts of the nephron and their function. With the BOWMAN'S capsule, the function is ultrafiltration. Ultrafiltration, which I said last time is to filter by pressure. Yeah, ultrafiltration is filtration by pressure. And I explained that I said blood comes in through the afferent arteriole, which is larger in diameter than the efferent arteriole. And then when it comes here, it is because it is going to squeeze itself through the small lumens of the capillaries here of the glomerulus. molecules that are small are going to be pushed out, much like what happens during tissue fluid formation of the normal circulation. So in this case here, small molecules are going to be pushed out, but more on that later in this lesson. Talk about the adaptations of the glomerulus. Says it has a high blood pressure that forces the small molecules out of the glomerulus, and we explain and say this is because the afferent arteriole is wider than the efferent arteriole. Afferent is where blood comes in, efferent is where blood leaves the glomerulus. Then we said it has many capillaries that give it a large surface area for ultrafiltration, and it has a semi-permeable membrane. The capillaries have to be many so that we can easily filter out as many molecules as possible. And then the semi-permeability of the membrane also helps us so that molecules can actually go through. If it was impermeable, we'd just have a mess happening there. But because the capillaries are permeable, or what we say semi-permeable, some molecules can go through depending on size. The filtration that happens in the glomerulus happens entirely on a size basis, not importance, not quality, not nature of the material. It happens on the size. Small molecules filter through, large molecules like proteins and red blood cells do not filter through the remaining blood. So in that case, we are going to have most of the molecules that are important, even the wastes, are going to be filtered through. That leaves the rest of the part, the remaining parts of the kidney to one particular function, and that is reabsorption. More on that later today. And then we talked about the proximal convoluted tubule. The proximal convoluted tubule has a simple function. The proximal convoluted tubule is the site for reabsorption of materials useful to the blood, like glucose, amino acids, water, minerals, salts, and so on and so forth. Then the loop of Fenley has one important function, and that is to concentrate the renal fluid. So you're saying here it makes the tissue fluid in the medulla to become more concentrated than the glomerular fluid. I'll explain that when I begin talking about the process of reabsorption. Then we talked about the diastole convoluted tubule is also responsible for reabsorption of chloride ions. Then the collecting duct conveys what now is called urine to the pelvis of the kidney, from where it goes to the ureter and so on and so forth. So we talked about these adaptations of the nephron very quickly. The nephron has a thin membrane for easier diffusion of materials, has a microalveoli to increase the surface area for reabsorption, numerous mitochondria. The thin membrane, remember, the thin membrane reduces diffusion distance. Okay, so most parts of the nephron have a thin membrane for easier diffusion of materials. We have a microalveoli to increase the surface area for reabsorption, numerous mitochondria which provide energy for reabsorption. Okay, so this is where we stopped. The stages of urine formation. Urine formation has two stages. The first stage is ultrafiltration, and the second stage is reabsorption. Ultrafiltration, like I have already said, is to filter using pressure, using excessive pressure. Okay, and then reabsorption simply means what you hear it mean, to absorb something back into blood. We are going to reabsorb some of those molecules back into blood. So what we need to know about ultrafiltration, the first thing is blood enters the glomerulus from the afferent vessel with a high pressure. Blood enters the glomerulus from the afferent vessel with a high pressure. Why? Because the renal artery is directly connected to the aorta. The renal artery is directly connected to the aorta, implying that the pressure that blood comes with from the heart is the pressure that is going to be moving with that blood, even as it enters the renal artery and it divides into the afferent vessels. However, when this blood enters the afferent arteriole, the size of the lumens change. This means what? That the amount of blood that is entering at any particular moment. So here we are saying blood enters the afferent arteriole at a very high pressure. Blood enters the afferent arteriole at a very high pressure. However, less blood leaves the glomerulus than the blood that enters the glomerulus. Why is this? The reason is that the diameter of the lumen of the glomerular capillaries and the efferent arteriole is extremely small, meaning blood has to push its cells through the narrow vessels that are in the glomerulus and the efferent arteriole so that it can go through. What happens? Because it has come at a high pressure, we are going to have a pressure buildup inside the glomerulus. This pressure buildup is going to later cause small molecules to be pushed out through the permeable walls of the glomerulus. It is going to cause small molecules to be pushed out through the permeable walls of the glomerulus. And as they are pushed out, they are collected in the BOWMAN'S capsule, and then they are filtered into the proximal convoluted tubule. So let's go back here and see what we are talking about here. So you're saying blood enters the glomerulus from the afferent vessel with a high pressure. However, more blood enters the glomerulus than that which leaves at any one time. So this high pressure that is going to be generated because of the continuously coming in blood and the little blood leaving, the high pressure is going to force small molecules out of the capillaries into the BOWMAN'S capsule. This is going to form what we call the glomerular filtrate. It's going to form what we call the glomerular filtrate, this one here. It's going to form what we call the glomerular filtrate. Now, this filtrate contains very many things depending on size. Every small molecule will be filtered out. So we have molecules like glucose, we have urea, we have water, we have salts, we have vitamins, and so on and so forth. As long as the molecule is small, that molecule is going to be filtered out. On the other hand, large molecules like proteins, the red blood cells, the leukocytes, the white blood cells, water, okay, some of these molecules do not sorry about this one, water is filtered out. Molecules like proteins, red blood cells, white blood cells, which are large, are not filtered out. Why? Because the filtration that happens in the glomerulus happens because of a pressure difference rather than happening because of importance. So filtration in the kidney does not happen depending on the importance of the molecule to the blood, but it happens because of the size. Now, because we lose important molecules during the first stage of filtration, we have to reabsorb back most of these things back into blood until we filter out only the waste products, urea and excess water. So the next stage is what we call selective reabsorption. The next step of urine formation is what we call selective reabsorption. Selective reabsorption starts after the immediate formation of the glomerular filtrate. And the part next to the glomerular filtrate, so the part next to the BOWMAN'S capsule, is the proximal convoluted tubule. So the first stage of reabsorption, selective reabsorption, happens in the proximal convoluted tubule. What happens? So most of these materials are going to be absorbed back by active transport. That is why we say that the proximal convoluted tubule, one of the adaptations we talked about in the proximal convoluted tubule was that it has numerous mitochondria. It has numerous mitochondria. So these mitochondria provide energy that is going to be used in the active transportation, in the active transport of molecules. So molecules like glucose, molecules like vitamins, some mineral salts are going to be pumped back into blood at the proximal convoluted tubule. Now, I want you to have this in mind. The nephron is not a standalone organ, a standalone tissue. It is engulfed in capillaries. The whole nephron is surrounded by capillaries. These capillaries surround it to quickly pick up any molecules that are being pumped away from the nephron. So these molecules that are pumped from the nephron are going to be pumped directly into blood. Also, molecules like sodium chloride and some water can be reabsorbed by diffusion. So in the proximal convoluted tubule, we have the reabsorption of very important molecules like glucose, vitamins, mineral salts, and some water. Some of them are absorbed back by active transport, and others simply diffuse back into blood. into blood. The second part after the proximal convoluted tubule is the loop of Henle. So what happens is, as this glomerular filtrate passes through the proximal convoluted tubule and it begins to go down the descending limb, water is reabsorbed back from the blood. Let me first put it so that people can see what I'm talking about. Okay, so here we are. Let me get a pen here. So this is the descending limb, and this, no, let me erase that. Okay. So this here, I'm trying to drawing using a mouse is not easy. Oops. This here is the descending limb. I'm shading it red. And then we have our ascending limb. Let me shade it blue. This is the ascending limb. Okay, now from here, get another color. Green. Let me get green. So as the glomerular filtrate is coming from here, the proximal convoluted tubule, it is coming. What is happening here is that molecules like glucose, let me get a pen here. So molecules like glucose, you guys, this is terrible. Molecules like glucose are going to be reabsorbed. Are you together? Molecules like glucose are going to be reabsorbed. And molecules like water, molecules like mineral salts are going to be reabsorbed. Now, when it comes to descending limb here, where I'm erasing now, water is going to be reabsorbed back into blood, making this solution here to become more concentrated, which is in this inside here. Making this molecule here, whatever is inside here, to become more concentrated. Why? Because water is being taken out. Water is being taken out. Now, when it reaches here, when it reaches the ascending limb, which is this one, the ascending limb, in the ascending limb, we have salts moving into the medulla. Now, salts are moving into the medulla. Remember, water moved out first, making the solution concentrated. Now salts are moving out, making the solution to become dilute again, but it makes the medulla to become concentrated so that more water can leave again. Let me repeat that so that people don't miss out. Okay? So we are saying on the descending limb, okay, as water moves out during the descending limb, okay, on the ascending limb, salts are moving into the medulla. Salts are moving into the medulla, making the medulla to become more concentrated. Now, when the medulla becomes more concentrated, more water is again going to leave via the descending limb. And because water is leaving via the descending limb, the glomerular filtrate is going to become concentrated. Okay? So because the glomerular filtrate is becoming concentrated, the ascending limb again, salts are going to be pumped out. So what do we know about the loop of Henle? In the loop of Henle, we know that the descending limb is permeable to water, but the ascending limb is impermeable to water. We also know that the descending limb is impermeable to salts, while the ascending limb is permeable to salts. So what happens is, while the descending limb is losing water, the ascending limb is pumping salts into the medulla, not into blood. It pumps salts into the medulla, making the medulla, which is surrounding the loop of Henle, to become very concentrated. When it becomes concentrated, more water is going to move out by osmosis from the descending limb. This is going to make sure that we gain back most of the water that we lose, because we cannot afford to lose too much water. So then we go up here to the diastole convoluted tubule, where we can again absorb back salts. We can absorb back salts, and then when it escapes, by the time the glomerular filtrate reaches the collecting duct, it is now fit to be called urine. It is now fit to be called urine. So I repeat one more time. Blood comes through the afferent arteriole. It enters here into the glomerulus. More blood enters the glomerulus than the blood that leaves, because of the difference in diameter. Because of the difference in diameter of the blood vessels, that is, the afferent arteriole is larger in diameter than the efferent arteriole. Okay? And then, because of ultrafiltration or filtration by pressure, blood is going to be filtered, and we are going to get what we call the glomerular filtrate concentrating in the BOWMAN'S capsule. Okay, from the BOWMAN'S capsule, the glomerular filtrate is going to move into the proximal convoluted tubule, where molecules like glucose, water, vitamins, mineral salts are going to be either pumped back actively, or they are going to be absorbed back by diffusion or osmosis. When the glomerular filtrate reaches the loop of Henle, on the descending loop of Henle, water is going to be reabsorbed back in blood, but on the ascending loop of Henle, we are going to have salts being pumped into the medulla. All salts are going to diffuse into the medulla. When they are pumped into the medulla, they make the concentration of the medulla to become concentrated. I hope you have not forgotten where the medulla is found. So they make the concentration of they make the medulla to be concentrated, such that more water is going to move out of the descending limb of what? Of the loop of Henle. So from the descending limb of the loop of Henle, water is moving out, while on the ascending limb, salts are being pumped into the medulla. From there, the glomerular filtrate moves to the diastole convoluted tubule, where more salts can be pumped out depending on the need of the body. We are going to see later that the osmoregulatory functions, we have something, ADH, which can affect the permeability of the diastole convoluted tubule. Yes, sometimes my subtitles will not be okay. Okay, so from the diastole convoluted tubule, we are going to have urine forming into the collecting duct, where it is going to be transported back into the pelvis, and from there it is going to go to the ureter, and then we shall have the urinary bladder, and that is how urine is going to be formed. Wonderful. So let us go through what we have as notes. So we are saying most of the food materials in the loop of Henle, as the filtrate flows down the descending limb, water is reabsorbed back into blood. So we are saying as the filtrate flows down the descending limb, water is reabsorbed back into the capillaries by osmosis. Okay? And then during the diastole convoluted tubule, I have explained how water is reabsorbed back into the capillaries when we are talking about the loop of Henle. So we are saying in the diastole convoluted tubule, we have the selective reabsorption of salts by diffusion. And then in the collecting duct, sometimes we can have water being lost. And then the remaining filtrate is what we can call urine. This goes via the ureter and is temporarily stored in the urinary bladder. So when the urinary bladder is full, then you are told, the body signals to you to go to the toilet, and then you are able to excrete. So the next time we shall be talking about excretion in plants, which is quite simple. Actually, let me give it as an assignment. Find out Find out how plants carry out excretion and why they don't need excretory organs. Say it, find out how plants carry out excretion and why they don't need excretory organs. Okay, I hope we have been understood. Thank you. Are there any questions? Any questions? Hello?
Yes, teacher.
Any questions?
Nara, but we just want to say thank you, but
Yes, yes, Nasasida.
teacher, thank you, but we are not in the channel. We are unable to access your notes.
I've not yet sent the notes. When I send them, I'll tell you. Have you been seeing the work?
Yeah, I have been seeing these tests.
Okay. Any questions? Any questions? Naruoga Victoria, say something. Okay.